basics of wine-associated microbes, and I put them into three main categories: bacteria, yeast, and mold. This phylogenetic tree gives you an overview of where each of these microbial types fits into the tree of life. Obviously, the bacteria, like lactobacillus and acetic acid bacteria, fit into this particular domain called the bacteria. And then in the eukarya would be the yeasts and molds. And this is just an overview of the microbes based on their shape and whether or not they have a cell wall, nuclear membrane, and their average cell size. The average cell size isn't particularly instructive because sometimes microbes will change size depending on the nutrient conditions of the environment. But as a general rule, the fungi, like yeasts and molds, are much bigger than the bacteria. Some of the common shapes of bacteria, with reference to wine micro, these are the main types: rod shape or bacillus, and cocci, that you can see here in two different types of arrangements, in a string of pearls type arrangement or a strepto, and then a staphyla type arrangement for like staphylococcus. All these organisms, at least ones that are important in wine microbiology, have a rigid cell wall. And because they are prokaryotes, they do not have a nuclear membrane. And they generally range in size from about 1 to 5 microns. The fungi, as you can see here, the first group, the cellular fungi called yeasts, do have a rigid cell wall. They do have a nuclear membrane, and that's why they belong into the domain eukarya. And they're, again, bigger than the typical prokaryotes, like bacteria. The fungal types that are more filamentous include the molds, and so that's what you see here. So this cartoon represents a mycelium, which is a group of hyphae. And so each individual hypha, one little filament of a mold, is pictured here, and then you put a bunch of these hyphae together and they form a mycelium. So again, the basic shapes and arrangements of bacteria, they can be spherical or cocci, as you see here, and so some are in groups of 2, so we'd call those diplococci. Some are in a string of pearls type arrangement that we call streptococci. Some have a more interesting arrangement, like this little group of sarcinae, which is just a group of 8 cells and packets of 4, 2 packets of 4, and then some are tetrads, and you can see the micrographs over here that kind of illustrate these arrangements, and then of course the staphylococci, which again have that grape-like cluster. Staphylo is a Greek word for grapes. So the bacterial shapes that are represented by the rod-shaped organisms, or bacillus-shaped organisms, you could be single bacillus or a diplobacillus, again, 2 end-to-end bacilli, or streptobacillus, again, sort of like a links of sausage. And so you see a classic streptobacillus like this lactobacillus species here, and then a classic single bacillus like Acetobacter. The Gram reactions of microbes are typically either positive or negative. Some microbes don't really some bacteria don't fall into either of these categories, but in terms of wine microbiology, all the important wine microbes fit into either one of these two categories. So the Gram positives are pictured here on the left, and this cartoon goes along with the actual electron micrograph that you see here. But we'll follow along with the cartoon because it's a little easier. The Gram-positives have a thick peptidoglycan layer or cell wall. They have a small little periplasmic space in between, and then this layer here is the plasma or cell membrane. The Gram-negatives are a little bit more sophisticated. They have an outer membrane, and then a periplasmic space, apparently a larger periplasmic space, but I don't know if anybody's ever really measured it. And then they have a very thin peptidoglycan layer. So you can compare that to the peptidoglycan layer of the Gram-positives. And then, of course, the cell membrane or plasma membrane is pretty much the same between the two organisms, or types of organisms. And, of course, we'll go over Gram staining in the lab portion of the class. I actually posted a video, and hopefully you remember that from Micro 221 or 224, whichever one you took. So this is a closer representation, close-up representation of a gram-positive. And so you can see some of the main features, these teichoic acids, for example, and then lipoteichoic acids, which kind of embed in the plasma membrane and stick out into the world. And each of these types of structures is essential for binding to surfaces, for example. And again, you can see the relatively thick peptidoglycan layer and get a sense for how it's constructed. And then here's a small little periplasmic space followed by a plasma membrane. And then embedded in the plasma membrane are various proteins that do all kinds of things. Imagine if this cursor is a nutrient that can percolate through the cell wall, and then they may have a pore in the plasma membrane that could then take that nutrient in. The gram-negatives, again, are a little bit more sophisticated. They have this more complex outer membrane, which is basically a partial lipid bilayer. So if we look at the gram-positive, you can see the full lipid bilayer. This is a lipid layer, this is a lipid monolayer, and another lipid monolayer interfacing to make a lipid bilayer. And so the gram-negative outer membrane is partially a lipid layer, and then the outer layer that you see here is lipopolysaccharide, which is indicated right here, lipopolysaccharide. And these are little sugars that stick out into the world and give microbes certain characteristics, like the ability to confuse your immune response, for example, by changing the composition of those sugars. Or they can also prevent the uptake of certain nutrients or prevent the infiltration of certain compounds that might be detrimental by just modifying the charge of these sugars. The other thing to notice here is that they have porins in the outer membrane, and porins, again, can allow, you can see they're like these triple-barreled transporters that can allow small molecules like amino acids and so forth into the cell. And once those amino acids get into the periplasm, they can then move through transporters across the plasma membrane, for example. Here's the representation of the relatively smaller peptidoglycan layer in the gram-negatives, and then, of course, the plasma or cell membrane. And here's just a cross-section of a bacterium sort of sliced in half and folded open so you could take a look at it. Many microbes have a capsule, which is sort of a sugar layer around the bacterial cell. And in the case of wine micro, capsules having microbes with gummy capsules potentially could influence mouthfeel and things like that in a wine. Whereas in addition to that, microbes also have you see the cell wall layers, and they have, I'm trying to look at the fimbriae, here they are, the fimbriae. The fimbriae are also essential for, just like the lipoteichoic acids and teichoic acids are essential for binding to things. So microbes can use fimbriae to stick to the enamel on your teeth, or a rock in a stream, or the lining of your gut tract. Inside the cell, there are inclusions. The microbes can actually store nutrients in these inclusions and presumably hang out for a while in nutrient-poor environments, as long as they have stockpiles of nutrients in these inclusions. And of course, some microbes have flagella for swimming. And here's our nucleoid. Again, we call it a nucleoid, nucleus-like, because there's no nuclear membrane around this. And so the DNA is sort of freely floating about in the cytoplasm. And of course, ribosomes typically stud this cytoplasm as a means for protein synthesis. Here's a picture of a gram stain of yogurt, and so you can see the lactobacilli, for example, the gram-positive lactobacilli, they've stained purple, and then the gram-positive streptococcus that we typically use to make yogurt. And here's a cloud, this pink cloud. Here's a cloud of milk proteins. Just a little bit about molds. Molds are great because you can actually identify them, at least a genus, just by looking at them under the microscope and recognizing different morphologies. You can't do that with bacteria. I happen to know that this is lactobacillus because I did some additional work to figure that out. But And I would also know that this is Streptococcus for the same reason. I can isolate the microbes and do some biochemistry or genetics to identify them. Whereas here, you can actually look at these microbes, these molds, and because of their sophisticated structures, you can kind of distinguish them based at least on genus. I couldn't tell you the species of this penicillium, for example, but I could tell you that it's penicillium. They have very distinct conidia, which are external reproductive spores that typically are piled in a row at the end of these little fingers, or phyalides that they're called. Whereas Aspergillus, when you look at these guys under the microscope, have a very pronounced vesicle, and then the spores radiate out of a vesicle. So again, I could tell the difference between Aspergillus and penicillium just by looking at them. This cartoon gives you a little bit more detail of Aspergillus and penicillium. And so you see the vesicle I just mentioned, and then the phyalides, the little fingers, and then the rows of conidia. The phyalides in penicillium are similar, and the conidia are similar, but there's no vesicle. The other thing to notice is that the conidiophore, the hypha that sticks out above the medium, is septate in penicillium and is not septate in Aspergillus. Another important mold in wine microbes, botrytis. This is in this particular case, you're seeing a bunch rot or gray mold as a result of botrytis overgrowing on these grapes. The conditions were probably wet for a prolonged period of time, and so this would actually be detrimental. But these guys can also produce something called a noble rot if the conditions change, in which they initiate wet and then you get a little bit of growth of botrytis, and then if the conditions turn dry, you can manage the botrytis and actually get something called noble rot, in which the microbes, in which the mold actually concentrates the sugar a little bit, and so you could use them to make a sweet wine like Riesling. That's something that we would call a botrytized wine, for example. And so here's a botrytis under the microscope, and so you can see this bunch of conidia. This cartoon here illustrates the life cycle of botrytis. And so here's a representation of botrytis as a bunch of conidia, and the conidiophore itself that you can see, and again it's septate, so there's little cross walls there. And so these little spores are released into the world, can infect a berry and then germinate, and then you see the hyphae penetrating the berry of this particular grape. And then eventually they would cause a gray mold or a bunch rot if, like I said, if conditions continue to be wet. And this is basically the summer cycle, where they're the happiest in growing in these big bunches. And then as the temperature changes and it gets colder, they would produce these resistant spore types, or even some of their broken off mycelia that would fall into the leaf litter and form the composition of the soil. And basically, but in either case, these things can overwinter, and they're pretty rigorous and tough, so they wouldn't necessarily be destroyed by usual conditions. And they can actually germinate into new fruiting bodies, essentially, conidiophores with conidia that can then go back out into the world and wreak havoc or cause noble rot, depending on the conditions. And this image is just showing you the mold life cycle, with some beginning with an asexual spore. And so that's here, the conidia, these asexual spores. And so a spore would germinate, and you can see the hyphae coming out of this guy. And remember that a massive hyphae would be a mycelium, and then the individual conidiophores that rise above the surface, as you see here, would then produce these fruiting structures that would then produce even more conidia that could go out into the world and colonize new surfaces. So like when you see a moldy piece of cheese in your refrigerator, and your instinct is to cut this part off, the part you can see, but the vast majority of the mold is still subsurface. So cutting off mold from certain things like that just doesn't exactly do the trick. This slide just represents the extracellular digestion of a mold. And so we use this basic concept when we make mold ripened cheeses, for example, like blue cheese or camembert or brie. And so again, you would make the cheese in the same way, but then dust it with conidia from specific kinds of mold, and then the conidia would penetrate. I'll go back. The conidia would penetrate. I'm sorry, the hyphae would penetrate the cheese, for example, and then the mold would produce digestive enzymes that could break down whatever the substrate is. In this particular case, it's fats and protein and cheese, and then create the consistency that's desirable, for example, in brie. Just a bit about controlling molds. Obviously, we can, and I'll get into this more at a later time, but fungicides are probably the preferred mechanism. And obviously, before it rains, so you're not diluting them or causing any extra problems with distribution in the wrong places. And heavy fog, also, that heavy moisture, again, can dilute and distribute in places that you don't want. Sanitation is obviously important by quickly going through and pruning and removing diseased fruit, and then of course moisture management, where you're you're using things like drip irrigation rather than just spraying water all over the place or indiscriminately. So drip irrigation is a good way to go, or tilling in organics into the soil to facilitate breaking up the clay or whatever kind of material is for greater penetration of the water.